Okay, let's dive right in. It's maybe the biggest question we ask ourselves, isn't it? Are we alone? Well, forever we've looked at the stars, but lately our eyes are really fixed on Mars.
Yeah.
The idea of life there, not ancient fossils, but life right now, it's huge. Finding it could, it could completely change everything about our plans to maybe colonize Mars. Might even stop them. So this deep dive is about that urgent search. It really feels like a race against the clock before we, potentially mess things up by going there.
It really is a monumental search. And what's so interesting now is this specific tool, the Agnostic Life Finder, or ALF. It's a compact, specialized. We're going to look at this new paper by Christopher Temby and Jan Spacek. It gets into how ALF might just be our best shot.
Right. So Mars, it always comes up as the place we'd likely go first. Setting up a colony beyond Earth is relatively close, has some potential. But that very idea, humans landing there, makes this search for existing life just incredibly time-sensitive, doesn't it?
It absolutely does. There's this critical window. It's closing. Scientists are rushing, basically, to see if life totally separate from Earth exists on Mars now. Because the minute we land, bringing our spacecraft, our microbes, even trying to be careful, well, telling native Martian life apart from Earth life could become, essentially impossible. This is really our chance to find out before we potentially contaminate the whole scene.
Yeah, imagine finding something amazing and then always having that doubt. Do we bring it with us? That urgency, it explains why tools like ALFs are getting so much buzz. So what's the big deal with ALF? What makes it different?
Well, ALF's core idea is pretty clever. It looks for life based on fundamental biological signatures, specifically things called polyelectrolyte polymers. Think DNA, RNA, those complex charged molecules that carry genetic info here on Earth.
Wow.
ALF is designed to spot molecules like that, even if Martian life cooked up something a bit different.
Okay, but hold on. If Martian life started totally separately, why assume it uses DNA or RNA or even something similar? Isn't that a bit Earth-centric?
That's a fair point, and scientists have definitely wrestled with it. The thinking isn't that it has to be DNA or RNA exactly like ours, but the suspicion, the strong hypothesis, is that any life would likely need some kind of charged information-carrying polymer, something to hold the blueprint. It's a key concept in NASA's Ladder of Life Detection framework from a big 2018 paper. It's less about finding our kind of life and more about finding the fundamental building blocks, whatever they look like.
Okay, so ALF focuses on these information polymers. But Mars, it's not exactly bursting with obvious life signs. So a huge problem must be just finding enough of these molecules, right? Dilute signals. If there's only a tiny bit of life, how does ALF even pick it up?
Exactly right. And that's where ALF gets really innovative. It tackles that dilution problem head on. It uses a technique combining electrodialysis and nanopore filtration. Sounds complex, but the idea is neat. You take Martian soil, mix it with water, and apply an electric field. Small stuff like salts, potassium, magnesium, they pass through these tiny, tiny nanopores. But the bigger molecules, the polyelectrolytes we're looking for, they get trapped. They get concentrated in a chamber.
Ah, okay. So it's like a super fine sieve using electricity, boosting the concentration of the interesting stuff.
Precisely. It ramps up the signal so you can detect it, even if it started out as just trace amounts. But then there's this other challenge, also from that NASA framework. These polyelectrolytes, they actually break down pretty fast in liquid water. It's called hydrolysis. And here's the twist. The ALF team sees this breakdown not as a problem, but potentially as a really important clue.
A clue? How so? Because they degrade quickly.
Yeah.
So if you do find them.
You got it. If ALF detects these molecules, the logic is they must have been made recently. They couldn't be ancient leftovers because they would have degraded long ago in any available water. So finding them might imply they're still being produced, perhaps by living organisms right now.
Wow. Okay. That's a big implication. So a detection wouldn't just be a hint. It could be almost definitive proof of active Martian biology.
It potentially could be. Just think about what that discovery would actually mean. Not just for science, but for how we see ourselves, our place in the universe. It changes the whole narrative.
That really flips the script on how you'd interpret a signal. It's pretty amazing. But, like we said, the clock is ticking. Plans for sending humans to Mars, they're moving forward. Each year that passes, that window to study a potentially untouched Mars gets smaller, doesn't it?
It really does. And ALF, which got some initial support from a NASA ANAC grant, that's Innovative Advanced Concepts.
Yeah.
It's definitely a top contender for this kind of search. But you know how it is with funding? Budgets shift, priorities change, its future isn't guaranteed. So we're facing this intersection of incredible scientific possibility and, frankly, some big ethical questions, too.
Right. the drive to explore versus the need to preserve potentially.
Exactly. Do we push ahead with colonization, knowing it might erase the very evidence we're seeking? It's a tough one.
So wrapping this up, where does this leave us, the listeners, thinking about all this?
Well, it leaves us at a really crucial point in history, I think. If instruments like ALF get the green light, get funded, fly to Mars, scientists might actually get their hands on direct evidence, Martian DNA or its alien equivalent. That would be an answer, finally, to that age-old question. But if the funding doesn't come through, or we get there with humans first, well, we might lose the chance forever. We might never know if we're truly alone or if life's been waiting, right next door all this time.